So let me share screen. Let me solve this. And then I guess I can do my video. Oh, okay. Sure.
Yeah, thank you for being here. This is Daniel Cruz from the Laboratory Systems Medicine.
That's right.
In the med school. And he's done a lot of great work and he'll share some of his work with us today.
Yeah. Thanks, young man, for the invitation. Thank you all for being here. I planned this talk to be, let's say, a little bit on the short side. We'll see actually.
Oh, so this is what we see up to.
Oh, okay. So thanks for the heads up. Well, that's not exactly what I wanted. Okay. How about now? All right, perfect. Okay. Yeah, so as I was saying, I planned this to be a little bit on the shorter side, hopefully for questions. And if there aren't questions, then we all get to go out early, so works out. But I'll be talking about data assimilation in medical models and digital twins. If you're not familiar with digital twins, if you're not familiar with data assimilation, that's okay. That's sort of what I assume. This is work that I'm doing with Adam Knapp, who's actually on the Zoom call, and he's the one that's done most of the computational work, all the computational work essentially. So if you have any questions, Adam is hopefully also going to be able to help me answer the question.
Okay? Hey, everybody.
You're loud and clear, Adam. Okay, so just to give you sort of a brief outline, I'll start off by giving a background on data assimilation, again, assuming that no one's familiar with it. And in particular, I'll be talking about Kalman filters, which is kind of like the most common type of data assimilation tool that's out there. Then I'll get into how to do data assimilation on stochastic multiscale models. I work primarily on agent-based models. They're very common. If you remember a talk that I gave like a year ago, that's primarily my bread and butter, agent-based models. But I'll give a little definition. They're mostly computational. I'll also talk about microstates and macrostates. And then I'll get into our results. And so a lot of this is work that's still in progress. It's part of the reason why we want to make space for like feedback and everything else. But we're trying to do data assimilation on ABMs, and I'll give this very easy sort of toy example with wolf sheep predation. It's kind of like a lot of Volterra type model, and then something that's actually published on viral infection. And I'll talk about microstate synthesis and quantization, probably just a bit of jargon right now, but I'll explain what all that is later. And of course, we always want to acknowledge all the nations, tribes, and people on whose land we work and we study. Okay, so actually, let me see if I can move this. Nope, that's not going to help. Hey, there it is. Okay, cool. The trick was not to touch it. Okay, so if you're not familiar with it, the concept of data assimilation, in essence, is that if you have model predictions and if you have observations from your target system, we're going to try to produce some kind of optimal estimate of what the truth is of the target system currently and in the future, right? So it's to fix things the way that we perceive the system currently, and then how we're going to project out in the future. And the underlying idea for data assimilation is that we've got two sources of information, right? Observations and model predictions, and both of them are incomplete and noisy. So on the observation side, maybe only certain details are observable, right? So if you're thinking about, let's say, viral infection, maybe you can only really get the viral count or something else, but you're not really going to be able to like see exactly what's happening at the tissue level, what cells are damaged, which ones are not damaged. And a lot of this is also really noisy from the observation side because measuring instruments have a lot of noise, right? So there's only a certain degree of accuracy that you may get from doing like a blood sample or doing like a CT scan or something else like that, only a certain amount of resolution. So observations are incomplete and noisy. And then model predictions, as I assume most people are familiar with, because I think most people who do math bio, are also incomplete and noisy. So they're incomplete sort of by design. There's only certain details that we capture within our model. Usually it's almost always there's something that we're missing, something that we're simplifying, so on and so forth. And what that means is that we also get a lot of prediction errors, right? Instances where our model prediction deviates from reality, and then these errors propagate and they get worse. So the whole idea for data assimilation is giving these two sources of imperfect information. We're going to make some kind of optimal forecast, and then you usually, right, we'll feed that in back into the model, and then we do this kind of loops. From a historical perspective, this doesn't work. Oh, does it work? Okay, there we go. A lot of data assimilation is was developed in the context of numerical weather prediction. So if you think like hurricane prediction, everything else, here in Florida in particular, we'll see like those spaghetti models and everything. Those are numerical weather prediction models for a hurricane trajectory. They incorporate information from like buoys and from satellite images and everything else. And this is really where the cyclic part of most data assimilation comes in. The idea that we make a prediction of where, let's say, a tropical cyclone is going to go. We get new information, we correct that, and so on and so forth. So that's really the pipeline under which a lot of this work was done, I think, in the 70s and 80s. Now, there's different ways that you can do data assimilation. Here I mentioned some examples for like initial condition estimation, model personalization, so on and so forth. And really underlying these kind of different realizations is whether we do it once or we do it repetitively, right? So numerical weather prediction side, we're going to do it repetitively. For a lot of personalization, we'll usually just do it like once at the beginning, and then we'll call it quits, all right? So that's kind of the context. Now, from our perspective, what we're working on within the lab is medical models and digital twins. So there's a lot of work right now that's being done for biological medical model personalization, this idea that you have information that's coming from a specific patient that you want to then tailor to that patient, tailor the model to that patient, and maybe you get like improve predictions in that way. But then there's also this concept of digital twins. And if you haven't heard about it before, this is like a big image from the National Academies of Sciences, Engineering, and Medicine. This is from a workshop that the National Academies ran on the foundational research gaps and future directions for digital twins. But in essence, a medical digital twin is an up-to-date model. And what that means is that you'll have the model, it'll change with respect to the patient. So you'll get information from your physical counterpart. It'll go into the model. Your model is generic in this very big context. So it could be mechanistic, like a lot of us are familiar with, statistical, it could be maybe AI, machine learning type of model, et cetera. But the idea is that you have your model, it produces predictions that help with decision making. So, help maybe a medical practitioner that informs decisions, then we get information, and then we have this sort of like continuous interaction. And then the idea here really is that the model is going to be changing with the patient, and it's going to inform the medical decisions. Very much in line, I should say, with a part of what happens with numerical weather prediction, right? So the models inform the forecasters advise different communities of what's going to happen, and then we get a new model update. The part that makes it a twin is the fact that we are getting this kind of like cyclic scenario. If you're interested at all in digital twins, I do recommend that you at least check out this report from those workshops. There were a lot of program directors that were present at those workshops, and now there's a lot of funding opportunities that have come out for that, or that are expected to come out for that. There was one that came out, I think, just like in March for mathematical foundations of digital twins in the context of like NSF's DMS directive. Yeah, I think so. So check it out, even if nothing else is sort of interesting about this talk. But this is in particular what we're working on, right? So in order to do this kind of data assimilation for really big, complicated models, we actually have to develop technology. Now, if you're talking about data assimilation in general, then you're really looking at the Kalman filter as your first sort of pass. And so the Kalman filter is a very old sort of data assimilation tool. In essence, it's an optimal estimation algorithm. It's optimal with respect to the fact that it minimizes the mean squared error of estimates. The way that it works is sort of highlighted here. This is from the wiki article if you go and check it out. But the essence is we start off with some kind of prior knowledge of the state. These will be our best guesses for where we're starting things off. And then the cycle is exactly what we saw before. We'll have a prediction step based on our computational or mathematical model. It'll produce estimate means and covariances based on whatever parameters you set up. And then we'll have the update step, which is currently just like a very nebulous sort of thing that incorporates the measurements. We'll have an improved estimate mean and estimate covariance, and then we'll repeat the cycle again. So that's it at a very high level, fighting most of the mathematics away. One of the things that I think is really the reason why people use the Kalman filter, even nowadays, is that measurements can be irregular. So this update portion where measurements come in, this is based on whatever measurements you have. So if you have like iterative measurements, you do this iteratively. If you have irregular measurements, so like maybe at 1 day, then 5 days, then 10 days, or something else like that, you'll repeat the prediction step without this kind of update and still be able to produce outputs, right? So that's one of the strengths, along with the fact that it's sort of this like efficient algorithm. But what makes all of this efficient and possible is this key assumption, which is that all the noise that you have with coming into your model and coming into your measurements is Gaussian. And of course, that's typically not true in reality. And so what's happened accordingly is that at this point, there's very, very many Kalman filter variants. I've listed some of them. We've got like the extended one, the unscented one. I won't go into what all those mean, but in essence, we're trying to like remove some of these assumptions for the Kalman filter, and in particular, the assumption that the noise is Gaussian. The one in particular that we work on is the ensemble Kalman filter. That's why I've got this one bolded. It's really good for very large models where you can't actually compute, or it would take a long time, I should say, to compute the covariances for all your parameters. So instead, what you do is you take a sample. And so you'll run, instead of just one loop with one estimate, you'll have an ensemble of estimates. And you can think of those as like the various spaghetti strings in your numerical weather prediction model. So instead of having just one trajectory for your hurricane, you have like 50 of them. And then that's your ensemble that you're updating iteratively. But I'm not going to get into the specific details, but that's sort of the idea there. What I will get into, though, is the technical details for the Kalman filter. And this is the most math-heavy slide that I have. This is actually getting into the specifics for how the original Kalman filter works. Okay? And there's some things that I want to highlight here. That's why I have these. I won't get into too many of the specifics. We can certainly talk about it if you have questions. But one of the main things that I want to point out here is that a lot of the update process ends up being sort of linear algebra that you're doing. And the Kalman filter historically was applied to ordinary and partial differential equations. So you have your ordinary or partial differential equations model that you then get certain matrices out of for your estimate means, for your covariances and everything else. And then you cycle through this process as you're doing everything with the assumption that it's like you have very, very many variables that you're doing your that you have in your differential equations model. But to sort of go through it again, we've got that prior knowledge that I mentioned before. The first thing, if you want to think about a first part, is this prediction step where we're going to be updating the estimated mean and then the estimated covariance. And then this here, like we'll estimate at time t sub k, given t sub k minus 1. So we're just looking at the past. And then here, you know, F is going to be your prediction model. So essentially you're piping in information, you're getting prediction. This little part that I grayed out is actually control. So underlying a lot of this is actually a lot of interest within control. So that's more so not the numerical weather prediction side, but in the robotics and engineering side. So they do a lot with like control associated to the mechanisms and everything else. We can focus on that here. The main thing that I want to mention for this prediction step is that we have this process noise covariance. So this is the noise that's coming in from the model that's sort of inaccuracy. And this is one of the things that we tune, quote, quote, with the Kalman filter, because we make an assumption for what that noise is, and that assumption actually has effects downstream of how well we're doing with our filter. So we'll do this production step, and then next, if you wish, is the update step. And then the main thing that's done here, sort of the key insight that Kalman had, is this Kalman gain, which I won't talk too much about the specifics for how this is formulated. The main thing about the Kalman gain matrix, though, is that this you should think about as your weight, how much you're weighing your model predictions versus the measurements, knowing that there's some kind of error that each of them have, right? And so we see this in this equation down here for updating the means. This is what's coming from my model. This is what's coming from my measurements, subtracting away the observation model, which I'll talk about a little bit. And then here, the Kalman gain is in the middle. So if the Kalman gain is sort of heavily weighting the measurements, then the model predictions won't matter as much, and vice versa. But the main thing about the Kalman gain is that we're expecting it to change over time. The intuition that you should have is that in the beginning, maybe we should really weigh the measurements a lot. We don't really trust our model predictions very much. But as time goes on, maybe our model does a lot better. And so we don't have to worry about the measurements as much. And then the noise we get from the measurements won't matter as much. But there's different sort of assumptions that you have here. Now, yeah. So the gain, as I mentioned, we should think about it as a weighting sort of between the model predictions and the measurements. In the simplest sort of way, you can think about it as like almost a 0-1, where like if I have my prediction and I have my measurements, if it's like closer to 0, then I will ignore most of this. And then I'll mostly be focusing on my model prediction. And if this is very close to 1, then what happens is I'll erase most of my model prediction and focus more so on the observations that I get.
I guess I was asking if you understand exactly what this gain is, then you know where it's from the first.
Yeah. So this formula comes from like a theorem specifically. I just won't get into I don't actually remember all the details for how this is formulated, but it's based on that sort of optimization idea. But yeah, and then maybe later, if Adam remembers more of it, we can talk about that. Okay, but one of the things that I sort of snuck in here is this observation model. This isn't really a mathematical model. It's more so indicating what information you don't see, right? So you have the truth of what the system actually produces, but then it's perturbed. Maybe you don't get a lot of that information. You can't measure every single thing about your system, and maybe you introduce some of the noise. So that's what this H represents. But what you'll see, even if I don't talk about the formulation, what you'll see in this Kalman gain is this R sub K term. This is the observation noise, and this is the error that we introduce here. So this is that other sort of tuning. We've got the process noise, which is the noise from the model. We've got the observation noise, which is this R sub K, and they affect how all of this is computed. And we have to choose that somewhat arbitrarily. So that's where the tuning comes in. Depending on the choices that we make, it determines how good or how not good we have. There's predictions we have. There's a lot of heuristics that are out there. As I mentioned, the Kalman filter is like fairly old. So what that means is that there's a lot of people who've developed like, you should use this strategy for this context and that kind of thing for tuning, but tuning is something that you inevitably have to do. So that's where, yeah, just emphasizing that this is the tuning portion that you have to do sort of inevitably. Okay, all right. So what we're going to be doing is applying, as I mentioned before, the Kalman filter to the stochastic multiscale model setting. As I mentioned before, it's really built originally for deterministic differential equations, which behave really nicely. We have a lot of tools for determining like the next step for a differential equations model, even if that's kind of a difficult model to resolve. At least we have some approximation techniques. But a lot of the medical models that are coming out these days are agent-based models. And if you haven't heard about it before, this is a computational model. And the idea is that you're simulating autonomous agents and you're trying to capture how these local interactions for these agents affect the system overall. So if we just look over here and we don't worry too much about the details just yet, ideally we should see that there's like these little circles and triangles and stuff that are moving around. They've done something to the environment. Now the environment, which had blue and yellow, is primarily gray and everything else. So these are the kind of interactions that we're seeing in this case. We'll talk about this model in a little bit. But typically, the ABMs that we work with are stochastic. Right here, you can see there's like a little bit of oscillation, a little bit of motion, a bit of noise. And they're also usually multiscale. So a lot of these agents will have internal rules. Sometimes those are differential equations or logic-based rules or something else like that. And what that means is that the state space, if you can think of a state space at any point in time for this model, is highly variable and high dimension. And it's variable specifically because you'll have agents. So here I've seen this like, for whatever reason, these little white agents appeared. your agents are born in the agile. So it's not like you even have one high dimensional state at every single time point. It changes based on what you add, what you subtract, and so on and so forth. So for this reason, typically whenever we talk about AVMs, we'll be talking about two different kinds of state spaces. We have the microstate space, which is a complete description of everything. So all the attributes for all of your variables, everything that you can possibly throw in at time t. So this is their full description. But usually in practice, you don't care about this, right? So if I had an AVM for viral infection, I wouldn't necessarily care about how many exact virus agents I have or how many exact epithelial cells I have and what each of those are individually doing. I care about summary information. What is the system effect overall? Is my host now very sick? Is it very healthy? That sort of thing. So for that, we usually work in the macrostate side. So this is a summarized description of aggregate variables across all the agents. So as I mentioned before, maybe you're doing agent counts, maybe you're doing quantity totals. If you have some kind of diffusion within the environment, you want to see what the total concentration is of some biochemical, something like that. The microstate is super variable, very tough to work with. The macrostate, we can fix the dimensionality. Maybe it's still really high, but we can fix the dimensionality. And a lot of work is done on actually trying to approximate a macrostate with differential equation models, with the stuff that I presented once upon a time, that sort of thing. So this is like really nice behaving. This is highly volatile. Yeah. So one can think of it exactly as a statistical description. That's a really good intuition for it. But so that's the kind of setup that we have. Now, the Kalman filter, as I mentioned, ultimately is just working with linear algebra. You're working with large matrices, but your matrices have to be fixed. So the essence that we propose is all described here. So this is how one might actually go about using the Kalman filter on such a like messy type of model. And I'll start at the bottom over here. So we've got the microstate level, then we've got the macrostate level, and at the very top, we've got observations, which first of all are sporadic, as we might expect them to be, and then they're incomplete, right? So we've got these agents. I've got a stationary one that's blue, then I got a magenta and a triangle. But maybe in my observations, all I can do is measure the yellow one, okay? So typically what I'll do is I'll run multiple simulations, right? And so I'll have multiple little microstates at time t sub k that I can then summarize with my macrostates. Here, the summary is very simple. It's just the agent counts, and they all happen to be the same. So the data assimilation process, what we're proposing is that we'll do the prediction by running the model. Then we'll summarize up to the macrostate level. And then here at the macrostate level, because it's a fixed dimension, we can actually apply the Kalman filter and we could resample as necessary. The resample is coming from the fact that we're doing an ensemble Kalman filter. So we'll get this measurement. We have the previous macrostates, and then we'll update it. And this will be, for whatever reason, the new version of the macrostates that we should expect. So this portion right here is, I would say, not too complicated. There's a lot of support for the Kalman filter. The hardest portion of all of this is the synthesis portion. And you can kind of think about it, right? If I give you agent counts, but then I ask you what should the microstate be corresponding to those counts, in general, I have very many, possibly infinite number of options, right? Depending on what restrictions I have. So if I've got like 8 blue, 5 magenta, 6 triangle, maybe I put all of the blue and magenta on one side, all of the triangle on the other side. Like that's a valid choice, technically. But maybe it doesn't really make sense with the previous runs that I have. Maybe this is the model state that I could never actually achieve when running the model. So the most difficult part of all of this is the synthesized portion. This is essentially a completely open question within this context of data assimilation. I think Adam and I and maybe one other person have both done it as far as I've seen in the literature. So it's pretty open. So this is where we'll do a lot of work. But just to sort of give you an idea for how this essentially functions, I'll just start off with a very simple wolf sheep predation model. And so this is the model in essence here. We've got, this comes from the NetLogo models library, but Adam re-implemented this in Python so that we could actually do any kind of analysis. And it's a Lotka-Volterra model. And so what I mean by that is we've got these 3 different agents. We've got wolves, we've got sheep, we've got grass. And then the way that they interact with each other is sort of like this really simple example. The wolves eat the sheep, they gain energy, they can reproduce, they die if they have no energy, they move randomly. The sheep do the same thing, essentially by eating grass, and then the grass, which is these little green portions, grows randomly according to some regrowth timer. And what you'll see is when you track the, I'll get into that in a little bit, but when you track the microstate agents, which are the agent counts, you have this lockable oscillation type of behavior that you would typically describe with differential equations. It's not exactly right because we've got a lot of the noise. There's a lot of random movement that we have happened here, but you do see when you pull back, you do see a lot of that kind of oscillatory behavior. So we wanted to look at this just as a simple example for how we might actually go about doing data simulation, let's say. And here I just mentioned that there's 5 model parameters, and in general, we may not know what the model parameters are. So sort of like if you think about setting up your own mathematical model for a patient, maybe there's some parameters for that patient that you don't know, but there's, but you have to sort of guess. So this is what it looks like if we just do random placement and random removal.
Yeah, so it's assumed that all agents are the same.
Oh, yeah. And they're sort of, so we seed them randomly and we have,
No, I mean, it's like all the sheep are the same.
Oh, yes.
But they're placed in different locations, but otherwise they have the same parameters.
Well, no, so they have an energy parameter, each of them, that indicates how much food they've eaten, and if they don't have enough energy, they die off. So that's like a little local rule that they throw in. But they all have the same variable. They have the quantity energy, but that quantity changes from one agent to another. Does that make sense?
They all have the same data structure and they all have the same model, but the contents of the data structure can be different. So like if you had 2 simulations, there's no way of there's no canonical way of pairing up sheep number 36 with a, in simulation 1 with sheep number 42 in simulation 2. They're indistinguishable in that sort of sense. Like there's not a canonical ordering or canonical pairing between any 2 different sets of simulations.
Yeah, what I would say, essentially. So for just one simulation that we did, so this is actually with a much larger environment, it's like a 255 by 255 grid. We just decided to try things out, assuming that our microstate synthesis was really, really naive. So if we needed more sheep, we just placed them at random. If we needed less sheep or less wolves or whatever else, we just remove them at random. And this is what comes out. Now, as I mentioned before, you can have different types of measurements. Maybe you can measure everything, maybe not. For this run, we assumed that we could just measure the wolf counts. And this is essentially how we do. So black is the true value. And we also have blue, which is this like average prediction. And then these bars that we have here are 1 standard deviation above and below. And because the model is really simple and it's very, very stochastic, we can see that in some instances, we're actually doing fairly well. Certainly, like the truth is within our standard deviation range, and we're kind of close, certainly here as well for the grass. Obviously a little bit noisy, but one would say that that's like within the bounds. The part that we don't do as well is even though we're still within the standard deviation bound, the prediction for our model is like spiraling outwards, and then the cone is just getting larger. So even though we have this within the cone, one would argue maybe that's not so. Okay. But one of the things, as I mentioned before, is that the parameters are unknown. So we assumed that we didn't really know what the parameters were, and we started off with an estimate. So while we're doing all of this, we're also doing the same Kalman filter update.
Yeah, is eventual extinction certain?
Sorry, eventual extinction certain? No. It does happen. So actually, the next example that I'm going to show we have some extinction, but extinction in general does not need to happen. You can have this kind of like oscillation where no agent goes extinct.
Oh, what I meant was eventually.
Yeah, so if you wait long enough, there is a steady state where the agents oscillate and they don't ever go extinct. But there are steady states as well where one agent dies off, or maybe both wolf and sheep die off and you only just have grass or something else like that. So you have multiple different steady states within your larger state space. Huh?
This comes into it, because typically if your states are bounded, then you expect how things to go.
Hmm.
But you go from there.
I think a part of that ends up coming up from the fact that you have only local feeding and that you have like this random reproduction that can happen. So sheep could like randomly produce an offspring sheep, and in this way sustain the population. And you can think about it if like the sheep are away from the wolves, then that can sort of matter. What are you going to say?
So I missed if you mentioned like your time scale here, so it's 0 to 1,000. Is that does that have units?
Oh, they're just arbitrary time for this kind of model. So it's like
Okay.
you can think about it if you wish, it's like day or something.
Right. So the true value that is.
Oh, the true value is this is what the actual agents were. I'm so sorry, I totally forgot this. So underlying all of this process, we ran the simulation once. We considered that to be the truth. So this is what actually happens. And then we hid the truth from ourselves other than certain measurements, which we perturb. And then we attempt to, using data simulation, attempt to approximate the truth.
Okay.
Yeah, so.
Okay, so like a synthetic data set.
Yeah. I forgot to mention that. So yeah, all the data we're doing is synthetic, but we do have those kind of assumptions. Thank you. So just like we have a trajectory that we pre-compute, we also have specific parameters that we set up. We don't have to worry about what these parameters mean just yet, although the naming sort of gives you an idea. But we also do this data simulation on the parameters. And you see like here, we're sort of doing like, I would say here we're doing relatively okay. We're undershooting. Here we're undershooting significantly. And then like wolf reproduction is actually not something that we ever capture. We worry about this kind of stuff, this parameter estimation, a little bit less. I'm not saying it's not important, but the main thing that we want to do from like a practical perspective is doing this state prediction, because this is the kind of thing that would be informative to the practitioner, right? They don't necessarily care about the parameters for our model. We do. They more so care about what the model predicts and what those outputs are. Okay. Now, on the note of extinction, one of the things that this does fail at is this example right here. So in this example, we're measuring wolf counts as before, and what ends up happening is that the wolves all die out. They don't have enough sheep. And so the Kalman filter does accurately determine that the wolves all die out. But within a wolf-free model, we actually end up having two steady states. We have this high sheep, low grass. This is what actually happens. This is the true value for the simulation. So we have a lot of sheep. We have a very little amount of grass. Or we have the opposite, where we have high grass and low sheep. And this low sheep is really no sheep, right? So if all the sheep die out, then we have no wolves, we have no sheep, and we just have a bunch of grass. We have like this prairie scenario. And then the error that we see here is that the Kalman filter chooses and commits to the wrong steady state. It thinks that all the sheep are going to die out and all the grass are, and we're just going to have a whole bunch of grass. And then it's really certain, right? So you can't really see, but there's actually like a little error bar, a plus or minus 1 standard deviation. And it is convinced that there's no sheep and that we should just have this constant amount of grass. So this is one of the problems that ends up happening. And I would say this is sort of like indicative of a problem in general with applying the Kalman filter to not just this context, but probably other contexts where you have extinction and you have other steady states that it can't really decide, that it has to decide on, instead of giving you some kind of like range saying like, this is option 1, this is option 2. That would be desirable, right? So this is an error that pops up. Yeah.
Is this related to model identifiability?
It is.
Yeah, it's not. So how do you?
So we have some ideas, not exactly for how to address that, but the ideal scenario when we're doing predictions, right, is that if we know that there are two steady states, or if we see that there are two possibilities, what we'd like to do is say, this is option 1 and give that as one option. But then for the other choice, say this is option 2. So maybe we can't tell you specifically which steady state you're going into, but we can tell you concretely that there's two steady states. So that would be informative, ideally, right? Especially if they're really, really significantly different. So that would be the kind of thing that's more so the thing that we're gunning for, as opposed to telling you always concretely which one you go on.
If you think in the mean field, then you should be able to tell which of these is stable. Does it pick the stable one automatically?
So that's a great question. As far as I can tell, and Adam can correct me on this, it does pick up the stable one, but we may have multiple stable options. So if you think about it from a mean field,
So in reality, if you were thinking you have two stable ones and some unstable in the middle, but they never see that.
that's exactly what I was going to say. So if you think about it from that perspective, we've got two attractors, and we're somehow riding that line, and based on that little bit of stochasticity, we can go one way or another. And this misses that subtlety altogether. It happens to zag in one way and then commit to that very concretely. So yeah, so that's an inherent problem. How long? Where I go to 11:30? Okay, and then questions, or is it 11:30 with questions?
11.
So we typically, the period ends, that's what I'm there.
Okay. Well,
We got to.
I promise not to.
oh, no. Questions are good. This is good. Okay, so I'll just mention very briefly that we have done a lot of tuning. This is again because those kind of different process stochasticity and observation stochasticities matter. For the sake of time, I won't talk about this too much, other than saying that what we're measuring is what is called surprisal. And surprisal, you can just kind of think about as the negative log probability of an event. And so here, the event that we're talking about for these surprisal measurements is the truth. And essentially, we're asking how surprising, based on the model prediction, should the truth be? And we're doing this for these very many runs. And so low surprisal is good. That means that the model is not very surprised to see the truth. High surprisal would be bad, which is like the model did not expect to see the truth incoming. And these little spikes downwards, this is when we are doing the data. So when we have data, we do the data assimilation, and we give the new prediction, as we might expect, the surprisal goes down. But the way that these all behave when we measure different things like grass, sheep, and wolves actually changes how well we're doing and how surprising things are. So if we're measuring grass and sheep, there's a point essentially where the model learns what the actual trajectory seems to be. And then on average, well, these are medians, but in general, it does fairly well at predictions. Whereas if we're measuring wolves, we seem to have, like this high surprisal, and it only gets worse if our process stochasticity goes up, if that noise from our model goes up. Hopefully, based on the previous slide, this shouldn't be too surprising. It's because we have these different attractors, and then we may reach an unstable state and then deviate, which for whatever reason is missed within the measuring grass and measuring sheep. So this is
Sorry, the surprisal, the log, the expectation of that would be the entropy.
I think so, right, Adam? Expectation of the surprisal should be the entropy.
Yes. That sounds right. Yeah.
Okay. Now, we did also try this out for a much more complicated model. So this is a published model for bioinfection. I won't get into all of the details, the one you were seeing before. In general, what we're seeing here, though, is a tissue and immune response to a generic viral infection. So we have a generic virus that's randomly placed at the beginning of this. And then we have your epithelial cells. If they're yellow, they're infected. If they're blue, they're healthy. And then the gray ones that you saw pop up once upon a time. So like out here, those are the dead ones, okay? And then we track a variety of things with this, like the virus, the cytokines, your immune response, all those little like colored agents over there are your immune cells. So this is like an actual published model where we have a lot more complexity, okay? And within this context, when we do this random placement and random removal, we don't do super well. I mean, we do well sometimes, but not super well. So the measurements are virus and cytokines. So if you're not familiar with those, just think like these variables here. Here I'm only showing 6 of them. We actually have like 60 some odd within the model, just has a lot of variables. But the main point here is that for some of them, we do super well, right? So total extracellular virus, this is dead on the prediction versus the reality. If I got total intracellular virus, which we don't measure, our prediction's still pretty good. But for other things like this total TNF, whatever that means, the prediction keeps thinking that it spikes up when it doesn't. And so you have this, tumor necrosis factor. Thank you. Yeah. So we have this thing spiking up, and then it has to keep correcting, but the model never seems to really learn what's going on. There's also other instances where like the model doesn't really know what's happening until it gets a bit more information. But we expect this to do poorly because we should expect that this like spatial information matters, right? If I've got different hotspots here and there, and I'm randomly removing and placing virus or randomly removing and placing immune cells, it's going to have a big difference, right? If I put all my immune cells randomly in one location, and then but my virus hotspot is in some other location, then of course, like my prediction is going to go off. So one of the coolest things, this was completely Adam's idea, that we decided to do is what is called quantization. If you're not familiar with that, it's essentially the mapping of a larger set into a smaller set. And you typically, when you have quantization, you track this quantization error. Without talking about the specifics, I'm actually just going to do a picture version. So this is the Mona Lisa in all its colors. And we imagine that we want to map this into just black and white. Okay? Well, we could just set a threshold for like what's black, what's white, and then round accordingly. The problem is that when you do typical rounding, you're going to get a lot of artifacts, right? So this is if I just round based off the threshold, we can pick up some features, but we lose almost all of this in the background. So this is if we haven't done this kind of thing carefully. But this is only one way that we can do quantization. Another way is what is called error diffusion. And so again, without getting into the specifics, the idea when you do error diffusion is you're going to scroll through your image, and you're going to keep track of how much error when you make your choice, how much error you have, and you're going to diffuse that error. There's many ways that we can diffuse the error. Here, the one that I've shown as you're scrolling, excuse me, is just, let's say you distribute it evenly across your neighbors. And then whenever you have that error accumulated, sometimes that means that instead of producing white as you might expect, you'll produce black or vice versa, because all that error accumulation indicates you should zigzag the other way. And so the product is this black and white image where you can pick up a lot more of the features, right? And so we're preserving a lot of the spatial structure over here and a lot of that information, even though we're only in this binary set. And I should mention that this, this works in general if you're going from a larger to a smaller. So this is 8 colors. What happens to the Mona Lisa then versus down here if you do this kind of error diffusion. And as you can see here, even though we've got a lot of noise, we can pick up a lot of the features from this. So this is the same kind of idea that we wanted to do. Yes. That's exactly it. Yeah. So the reason that I use this example is that this is a way that people do or used to do image compression. You go from like 64-bit to 8-bit or something else. Yeah. And you learn
anyone who used a Mac in like the 90s be pretty familiar with this.
Yeah, exactly. So just to give you an intuition for how this works within our context, let's imagine that we wanted to increase the number of healthy cells, okay? So what'll happen here is the first thing that happened is that we arbitrarily have increased the amount of agents that are healthy cells. And so you can think instead of like having one agent that's a healthy cell, now we have 1.2 agents, which doesn't make sense from our ABM perspective, but that's what we're going to diffuse out and correct out. Okay. And then as we go through using the error diffusion, if you see I know it's small. If you see here, we're sliding through. And what should happen is that the borders should get a bit fuzzier. And then this hole right here and here, for example, should fill in. So as I do this diffusion, and I just sort of scroll past quickly, we see that hole fill in. We see some of the other gaps happen and everything else. But in general, we don't have healthy cells just randomly appearing in the middle of nowhere. And so if you do a comparison of the original structure with the diffuse structure, the structure itself is very similar, even though we've added cells. Yeah. So essentially, but keeping in mind that what we want ultimately is a discrete number of cells. Because for a lot of agents, what we end up having. Yeah. But that's the idea. So this is in essence what we did. You can actually see a larger video of this later. But when we did this kind of microstate synthesis for addition, for placement and removal, we improved quite a bit. So the main one that I pointed out previously, TNF, Before we were sort of like oscillating all over the place, it wasn't really able to predict it well. Here, it looks like it takes a while to make that kind of prediction, but then eventually the average model prediction does a fairly good job. And then we haven't lost too much on the other ones that we were doing well. I should say that it's not like we're doing, like it hasn't fixed all our problems. So for like IL6, for example, the model still seems to need a really long time to figure out what's going on. And then for IL1, it's still having problems determining what's happening. This, I think, is probably because IL1 has dropped to 0 and the model doesn't think that it should be 0. So we have this kind of spiking process. But in general, this sort of first attempt does seem to correct a lot of the problems and improve a lot of the model predictions. And since it's already 11:30, I'll go ahead and just conclude. So these are the contributions that we have. We've got this algorithm in general for applying the Kalman filter, and we've got this approach based on quantization for microstate synthesis. As I mentioned before, though, this is like wide open. There's a lot of avenues to improve the predictions, generalize things. Adam and I are talking about what to do for handling those edge cases, maybe presenting something. I mentioned that a little bit. And then we also are investigating some alternatives for microstate synthesis. A lot of people are interested in machine learning. We figure like maybe that's something that we could try out for like machine learning, predicting the next model state based on the previous one, whatever that sort of means.
Also, the synthesis of the microstates is something that we're looking at doing with machine learning as opposed to the quantization.
Right. So thank you all for listening. This is the Lab for Systems Medicine. We're all the way out in the Department of Medicine, but most of us here are mathematicians, and this is Adam. So yeah. Are you going to questions? Yeah, I do appreciate that y'all asked during. Yeah.
I guess, I just have a very naive question, I guess. You mentioned that the model predictions, some of the figures that you showed, it thinks it should go a particular way, but at the same time, it captures a lot of the qualitative features. So if we were to take the average, let's say, smooth out the predictions, it seems like the match would actually be not that bad.
Yeah, so that is one thing that's true for sure, and that's what we're seeing when we do surprisals. So the surprisals we're doing over the course of like 1,000 runs of this, keeping in mind that there's a lot of stochasticity. So when we look at the surprisals, that's a lot better. We're still running those, so that's why I didn't have a figure for that. So some of those are not bad, but I think that the ones that sort of give me concern is when we seem to be doing really well and then we spike outwards, right? So it's still within the range. On average, we're probably still doing well, but that seems to indicate that like we're missing something, probably one of these like unsteady states that we've gone in the opposite direction.
The Ancock-Rel model that we're using as well, it has a lot of it has a lot of variability in the model itself. So like sometimes, for instance, you see simulation runs where IL1 doesn't get secreted at all. So
these observations are the consequences of negative feedback, trying to correct the model.
That's exactly what they are. So what happens is every single time you see them spike down, it's because we've gotten a measurement, and the measurement course corrects us, says, okay, this is actually where you should be. Yeah, exactly. And so when that oscillation sort of goes down, so like at the tail end of here, that means that we're okay. Now we're on the right track again. But it being really high means that you're sort of like still spiraling outwards, right?
So we can reduce time to convergence.
That's a great question. That's where we don't know. That's one of the open things that we'd like to look.
I didn't quite catch that. What was the?
Oh, is there a way that we can reduce the time to convergence for these sort of like spiky oscillatory instances?
Yeah, we would very much like to do that, especially with the parameters. Just to interject one thing that Danielle mentioned, but like I'd like to hit it again. Like some of this spiky behavior is also due to the fact that we have non-negative parameters, and when the simulation is actually at 0, the non-negative parameters, you can only really sample them, positively. Like that's, the vast majority of the time, you're going to get a positive sample. So one of the things that we're looking at is to try and develop something where, you can't just do a log transform, right? Because
So
0 is a legitimate value.
So what we're sort of getting at, just to add on to what Adam said, is like when we think of these like bounds and ranges, right? When we're doing the sampling and we're trying to guess what happens, if we don't think it's dead on 0, if we're not certain yet, then that's where we're spiking upwards, and that's where it's throwing off some simulations.
So the idea of Kalman originally was to assume that the noises are based out.
Yeah.
But that wouldn't work well with your non-negative things.
Exactly.
So what if you do the log? So what if you switch to the logarithms and do everything in that space?
So this is one thing that we haven't tried out, but that we have considered. One of the other things that we're thinking about is, what was it, multimodal Gaussian, Adam?
Yeah, mixtures of Gaussians.
Mixtures of Gaussians, something else like this. But we have thought about log normal as well, specifically to try to like fish some of that out. Yeah, but this is all sort of like first pass type stuff.
Yeah, so the log normal, so the problem is, so we have done this. We have done the log normal. One of the problems, of course, is that, for instance, in the wolf sheep grass, when the wolves really do go extinct, and you've got and you're doing things in the log normal, now when wolves are 0, the log normal of 0 is, negative infinity. And this really, really throws off anything that's based on like a Gaussian distribution in the log space. Because you can shoot to negative infinity really, really fast when the wolves go from 1 to 0. And so the thing that we've been talking about is dealing with wolves, positive number of wolves, differently than 0 wolves. Kind of not just doing a log normal, but like there's a log normal model and there's a wolf equals 0 model.
So it's both actually. So the parameters are, some of them can go to negative and everything else, but then there's also the problem with the state variables. So it's a both sort of thing. But I think we're out of time now, Adam.
Okay.
And thank you for all the questions. Oh yeah, so that's Adam.